Alright, so hacking really isn't solving problems for the fun of it. Really just getting down to the crux of it and hacking it, having fun with it, getting your friends together and solving shit, coming up with niche solutions. And so it's anything like that involving biology. Introduce myself. Okay. I'm Matt. I did a degree in microbiology at UNSW. Started up Biohack Sydney about 3-4 years ago, and that was just kind of getting the community together. Then following on from that was Biofoundry, which is the actual, which is the physical construct of it, the lab and actually getting our feet on the ground. And also work at the Michael Crouch Innovation Center at UNSW. It's a hackerspace, event space, just aimed at really getting universities into this kind of world. So, of course, with Biohack, as you heard from Meow, it's all about the community. It's relationships, it's where you can go, it's getting people from outside of biology and all coming together. And then we had to have the space, so we have this wicked lab in Green Square named Biofoundry, and it's in an artist studio with over 50 artists, and now comes the collaboration. And this is the LAMP Diagnostics team, you'll hear about it more, and we've got doctors and nurses, coders, and of course biology people. That's our lab during construction. So much better. This is a bio art course that we ran. So biohacking encompasses all aspects. It's not only the, solving a problem, it's also having fun with it. It's also playing around and getting people in there. Growing crystals, making cyanoprints. This was helped run by JJ Hastings, an amazing bioartist. This is the lab down in Melbourne called Bioacquisitive, and that's biohacking. Just getting a bread crate and having it as a glass drying rack. Like, we do this without any funding. This is all self-funded from the roots up, just building it ourselves. Pallets and everything. And we're trying to work into this shifting nature of work. So now you are your own independent asset. You are fluid and your skills can help in multiple groups, and basically we're hitching onto that. And yeah. And so that's like the concept of it. It's like bringing everyone together to actually tackle these things. And the actual, what people have done with it is democratizing science and bringing down the costs and just finding new ways of doing it. So this syringe pump usually costs a couple grand or something. They did it for under $100. That's a DIY CRISPR kit. CRISPR being the new fancy gene editing tool, which can really accurately splice lengths of custom DNA into a genome. And there it is. This guy is running an electrophoresis setup using 9-volt batteries and some foil. Electrophoresis is the separation of DNA through a gel by applying voltages on the other side, because DNA is negatively charged, and the DNA will run through the gel slower if it is larger. So you can separate based on size, and you use it for gene typing, you can do sex testing, you can test if your CRISPR experiment actually worked and it's expressing. It's one of the most used instruments. This is OpenRov. So we have a really good friend, Dom, and he works with this foundation. They make amazingly cheap underwater drones, and this is where it comes in. Like, it's not biological related at all. It's tech. It's software, hardware, but we use it for biological research. This is where bringing everyone together is really important. This is Peter Simpson Young. He and Neil. Yeah, him and Neil created the transcranial direct current stimulation device. They're incredibly cheap and doing actual experiments with it. And he is trained in this. He went to uni. He knows the science about it.
That's just sapping his head.
No, doing it well. This is another art-based project. This is from Symbiotica, and what they did was they routed audio through neural cells on a chip, and then outputted that, and did a live performance using it. Fucking awesome. These guys in the Netherlands, they went, hey, kelp, it's everywhere. What do we do with it? And they turned it into furniture and roof rafters and installation for walls, and it sequesters carbon, non-toxic, fireproof. And Chai Bio is creating amazing open source, really good quality lab gear that's, cost maybe $2,000, where before it was $20,000. So democratizing science, and they're making money out of it too. And yeah, so that is biohacking. Questions, please.
Yeah.
Ethereum. There's possibilities of incorporating with genetics, and so tracking your genome and securing it on the blockchain, someone was saying. But really that's for us to talk about later, I think. Does anyone have any ideas of how we could mesh?
Yes, you can put the Sci-Hub or something, the free data, scientific publications on the blockchain, so everybody can access it, not pay for the Elsevier or someone else.
Yeah, and once it's written down, and it's solidified, and...
Well, I was chatting with someone at the Department of Health at the blockchain conference before, and they were saying, looking into Ethereum and all this jazz, but I love the idea of you putting your genome on the blockchain, it's unalterable, it's immutable, it exists there, and you get to decide who uses it. That's
Superman.
Kind of fucking cool.
Immortality.
Yeah.
Definitely.
So someone can clone your
Yeah. That's right. Now we're talking.
There was also security around genetic material. So you could conceivably use the passkeys to then access genomic data, because genomic data is, of course, extremely sensitive. Yeah.
What about those people that putting those chips in their hands to do PayPass?
Yeah, definitely. Body mods are a very popular part of biohacking. Yeah.
So what's the ultimate aim of biohack?
The ultimate aim for biohack. I think really the spread of technology and to make it more accessible. At the moment, it's really sequestered and expensive to do diagnostics and to do G-mode agriculture and to implement all of these world-changing solutions. And so biohack is primarily about reducing the high level of accessibility into both creating these technologies and accessing them.
So you have a community and you organize events?
Yeah, so to create the community, we went to events, we went to other makerspaces, went to the Maker Fair, we run events, go to hackathons, everything about conjuring and bring everyone together.
Just on that, this is Biohack event that you're crashing.
Yeah. Welcome. Hey.
Yeah, is there a website for that kelp project?
Or has it got a proper name? Just Google.
Just Google it.
I would try and pronounce it. Rearda Big and Van Duxton. But I can post it up to the.
Yeah, post that.
Yeah. Yeah.
I'd love to hear some more information about the neural, sort of the music one.
Yeah. Yeah. So he got some. Pretty sure it was skin cells, then turned them back into pluripotent cells, then went to neural cells, and then planted them upon a chip. And then, depending on which neurons were fired,
Yeah.
it would then go through the neural blob and output something. I don't know how much they engineered the outputs. I think the key point of it was fairly random and just determined about how that organically grew.
That's crazy.
So where exactly? Are you sure it's skin cells that you used? Or?
It could have been.
So is it a German group or?
No, this was a Perth group. It could have been Ethereum or from Perth, Symbiotica. They're based out of the University of
So
Western Australia.
In the 80s and 90s, they used neurons from snails to and placed them on semiconductor surfaces. So it probably stems from that. I'm not exactly sure. So
Yeah.
that's why I asked whether you sure it was the skin or.
Yeah.
So it stems actually, I think, way back to that was and hence the Germans.
Exactly. And then it took how many years for artists to begin using it?
Oh, it takes a long looking at that.
Yeah.
So kudos for the presentation. I think as a biologist, I love it that 3D printing is going to enable me to do everything basically in my wardrobe or in my garage. My problem is with the bio stuff. I think the bio stuff is the expensive bit. So you can 3D print your dish and your microfluidics platform, but you can't print out the CRISPR system and the biology that is there. You can't print out the plasmids. That's the expensive bit. So if you want to do the PCRs, if you want to do the plasmids, if you want to do anything with gene technology, that is I have a feeling that's the bottleneck for the biohacking. So how do you guys cope with this?
That is definitely a bottleneck at the moment, reagents and etc. The way we're getting around it, we got a chemistry group into the same warehouse, and hopefully by working with them, we can generate new ways of producing those things cheaper. Yeah.
So all these amazing stuff that you guys have created or you guys seen, how are you commercializing it? Do you have a plan to do so? And do you have anyone following you guys around and kind of providing advice as to how to make it even cheaper and to eventually be able to turn an economic value, for example, out of these inventions?
Well, yeah, we definitely plan to. There's been a lot of companies in America who have received funding and have released products and are actively making money out of it. Here we're developing a new diagnostic device, and so we would sell that onto companies as well as the cartridges that are used in it. As for other commercializations, Meow's infant formula is a good example. We could have done that here if we had, we're up to date with America.
Right, but do you have people on your team or working with you guys every day to try to at least commercialize them or try to
No,
sell this?
we're too busy doing all the other stuff.
I think it's also really important to note that one of the key principles sort of biohacking is keeping a lot of stuff open source, like as much as possible. So while it's nice if you can make money from it, like you need to live, it's nice to be able to get some cash back. It's also important to us to keep it as open and accessible to everyone as possible, and not just try to turn everything into commercialize everything.
Yeah, so it's a spread of having your cash cow and having everything open. We also get funding by hiring the lab out to companies, and so we charge a premium if that is a established company using our lab and if you're just a student, we charge you a lot less. Yep.
I think one way you can raise money with your projects is to issue Ethereum tokens to projects. And people, if they like the idea, they can buy into it. So you can use it to fund it.
Yeah.
Is your model similar to how a lot of open source projects work in software, where they have kind of a lot of the base technology completely open source and anyone can use it, but you can pay for like support or help or
Yes.
like these other?
Yeah, so Chai Bio, the guys making the PCR devices, they firstly get their money from just building it for the people, because people using it generally don't have the time to assemble it, even though all the schematics and BOM is up there. And then they also get money for service and testing and supplying parts and reagents for it as well. Yeah.
What do the big companies say about you guys, or have they heard of you guys yet?
No, no big companies yet. Deloitte seems kind of interested. Apart from that, UNSW and MCIC very interested in what we're doing, and
You can name drop. Follow them out.
Primary healthcare.
Primary Healthcare is interested in our diagnostic device.
Big pharma company.
It's not really a pharma company, is it? It's James.
So,
It's not a pharma company.
No, we own like pathology companies and medical centers and imaging centers across the country. So Laboratory Pathology, that's us. Queensland Medical Laboratories, Western Diagnostics, Derbyshire Pathology. And then that's like the main labs. And then you got all the splinter regional collection centers and medical centers and imaging centers. We used to be Symbian, we bought them. It's like us and Sonic, we're like the two big ones in the country. So
Yeah, those guys.
yeah. And they're also, by the way, on the open source question before, when their thoughts on like this stuff, you think that they want exclusivity if they were going to fund anything, not even interested. They want what we want. They want a change they want this biohacking stuff to succeed. They want like stuff to change, and they're happy to throw money at it. So there's plenty of people out there who really want this stuff to work.
So when did Biofoundry open, and what did you have to do before that to do what you wanted to do? Like that's kind of the cool thing about Biofoundry that I liked when I first saw it. Like you have this idea of like a bio space, and you think like white rooms with like really nice equipment. And I went to Biofoundry, Meow showed me, and I was like, oh my god, this is fucking amazing. This is like Blade Runner type stuff. It's like he was telling me there's 2 machines in the background. They're $1 million each, but the UNSW was just like giving them away because they were upgrading. So it's like all this kind of like pillaged and borrowed equipment in one place, which I think makes it really grungy and awesome. And I feel like that's where innovation now and in the future is going to happen more so than like within university silos. So original question: when did it open, and what did you have to do before that to make?
It originally opened a year and a.
So we opened September of 2014.
2014.
15? Oh, 2014.
2014. Yeah.
And we opened our first space in Stanmore, in a 12 square meter. Was it 12 or 15?
Yeah, it was very
small space. We really quickly ran out of space. It essentially turned into storage. So we're on a hunt for a larger space. And that's when we moved to our current space in Alexandria.
Yeah. To get that done, we had to first start Biohack, get people that were keen enough, get people that were willing to actually come in and do the work and do all the things. We had to secure the lease, obtain the gear. Thankfully, universities are very quick to upgrade, and now we've submitted the paperwork to get PC1 status, and so that's when we become a lab that's able to do genetic modification, which is the next big step, and yeah.
So effectively, who's paying for these?
We are.
You're paying your own money. This is bootstrapping, is it?
Yes. So we are getting now some money from businesses leasing out the lab, and so reaching a near equilibrium.
So you initially started with an equity
Yes.
So we're incorporated as a non-for-profit charity, and essentially the founding members of Biohack Sydney and also Biofoundry bootstrapped essentially, but we're now almost, as Matt said, close to breaking even from the startups that are currently working in our space.
From the hiring side, leasing out the space.
That's right, yeah.
Okay, thank you.
Yeah.
I posted on the Biohack wall a while ago, but maybe some people didn't see it. I just thought I'd re-advertise it. There's a organisation called Falling Walls. They have a conference on in August 24th, and applications close May 23rd. And the idea is this is a venture capital firm, and they fund scientific breakthroughs and startups in that field. So people who are in Biohack, who have an idea for a business, it can be an open source idea for a business as well. I strongly encourage people to apply for it because I think you guys have got heaps of good ideas that could win this conference. So yeah.
May 23rd, Falling Walls. Where are they based?
No, I think it's a German institution actually. But so they'll have a conference in Australia in August, and then they go on to Berlin in November. So I posted up on the Biohack Sydney wall.
Cool. Awesome. Any more?
How much is approximately to lease to rent the space?
To rent the
Even
space?
if you're not established company nor student.
So far we have been just negotiating per person. So finding out how much they're using it and how able they are to pay for it.
Okay, so the number is
I don't do finances, Maria.
Can we talk later? So we've submitted our application for PC1, but we're yet to actually have obtained it. So we're not formally open to the public just yet. So, yeah.
And what kind of equipment do you have for gene splicing, like microarrays, something?
For gene splicing?
Yeah.
You don't really need too much equipment for gene splicing.
No, I'm just.
You can do it in your kitchen using that kit there. But we also have gear such as the PCR machines, which can amplify specific pieces of DNA. We have access to the reverse osmosis water filters, so you can get ultra purified water. We have so many centrifuges, it's not funny. And of course the laminar flow hood for sterile work. These other machines, automated liquid handler with inbuilt PCR, that's a Connolly pick and counter, and this is a gene chip arrayer. So you can stitch bits of DNA onto glass and then wash over your samples and see how much affinity they have to them, say. Yeah. Cool. Yeah.
How easy would it be to make a contagious disease?
They're based in Alexandria. Yeah.
I mean, you'll definitely be noticed because you'll be ordering weird primers and stuff like that. They captured like ISIS's head of chemical warfare recently, and he was, struggling to make advanced chemical weapons and could only do mustard gas or something. So, yeah, not many people really want to do it, and if they do, then they will be ordering really weird stuff.
Also, anything that happens in the lab has to be like submitted
Of
to
course,
us,
yeah.
and we have to approve that you're going to do that work. That's important.
Yeah, you can't just walk in there with a random bottle of something and start doing something.
Yeah.
And Meow's got the regulators on speed dial, right? Yeah.
Yes. Yeah, so we are working with the Office of Gene Technology Regulations. They seem to like us and want us to do good things, and they know that with us being around, it actually makes it safer. So.
Yeah. Hey.
So Sophie's going to do a quick talk on LAMP Diagnostics, which is our microfluidic qPCR cloud-based diagnostic machine aimed at home testing of STIs.
Each detector was a great name.
Yeah, cool. So yeah, my name's Sophie. I'm also from Biofoundry, and I'm just gonna quickly talk about a project that we've been working on, which is called LAMP Diagnostics. So, bit of background: the project started at HackerGong last year down at Wollongong Uni. Yeah, cool thing. So Biofoundry decided to send a team down. We found this paper online saying basically the entire open source specs of how to make a device for rapid and robust amplification of DNA so that you can detect bacteria and viruses. And we're like, that's really cool. And it's also like way cheaper than a normal DNA amplification machine. So let's go to HackerGong and make it. We started with, Matt and this bunch of electronics and spent a whole weekend basically soldering together a circuit board and trying to make it, with the end goal being rapid detection of STIs. We were hoping originally, we were like, we're gonna get it out to everyone's house, and then people don't even have to go to the doctor, they can just be at home and be like, I don't feel right, or, like regular testing. Turns out that that's still illegal, so now we're gonna aiming to get into doctors, and then to change the laws, long term. But HackerGong was successful. We came away with the best Internet of Things device hack, and the most innovative 3D print, and also
Internet of Things.
and there were three.
Yeah.
2, 3. So yeah,
we won the Red Bull.
Oh yeah,
prize.
Red Bull gave us a bunch of Red Bull.
Yeah.
Yeah, so we had a good weekend at HackerGong, but that was sort of a really, really basic prototype that we got going. So we decided that we want to keep going, we want to make this into an actual device that we can get out to people. So basically, I'll go back a few steps and explain what it is. So it's called LAMP Diagnostics, and LAMP is basically a way of amplifying DNA. So you guys might have heard of PCR, which is the traditional method of amplifying DNA, and it's got 3 steps. So you have to heat the mixture to separate the strands, and then the polymerase fills in, and then you go back down and you reheat. Basically, you gotta keep cycling through temperatures. And what LAMP does is it's isothermal, so it all happens at the same temperature, and it's got this really cool enzyme that can actually just smash through the DNA, break apart the strands, and fill in to copy it. There's meant to be a movie, but it's clearly not working. So, cool, we've got it later. Okay, so this picture is from the paper that we based our device on. We made a bunch of modifications from that, but the essential basis of it is that at the top here, you've got a microfluidic cartridge. So they use 96 wells. Ours is going to have a lot less than that. We're probably looking to have 4 or 5 inputs for samples, and each of them will go to 3 replicates for each sample. So you're going to put in hopefully raw samples, so either just urine if you're looking for chlamydia or gonorrhea, which are the 2 target organisms we're first aiming to detect. LAMP is really good because it's not inhibited by urea, so typically if you want to amplify DNA with normal PCR, the urea in your urine is going to inhibit that, and you need to do a whole bunch of cleaning up of the sample before you can use it. With LAMP, the aim is to be able to just use pure sample and skip that whole step. So you'll put that in, either with the reaction mixture, all the other stuff that you need to amplify the DNA, or in the long run, we'll hopefully actually be able to produce these cartridges with the reagents already inside in dried form, and all you'll have to do is add sample. But basically that will go in. That's sitting on top of an Indian titanium oxide coated sheet of glass, which gets heated up to 60 degrees for about an hour, which allows the reaction to occur. So if you've got chlamydia, gonorrhea, whichever bacteria you're looking for, then that DNA is going to be amplified. And as it's amplified, there's a fluorescent probe that gets inserted in between it. So the more there is, the more it's going to glow. And we measure that fluorescence with this array of phototransistors down the bottom. Blue LEDs excite the fluorophore, and then it goes through this greenish filter, and we measure what fluorescence comes through. So that's the basic premise. The device then will probably have an LED screen on it that will either say, yes, you've got it, or no, you haven't got it. Or if we want to add another level of complexity, we should also be able to make it quantitative. So it can actually say, you've got this many copies of it, you've probably got this many bacteria in your sample. You've got it really bad, or like, oh, just a little bit of chlamydia, still bad. Yeah, so that's the basic overview of the device. Where we're at, who needs it? We identified this need basically just for better testing of STIs. You know, at the moment you have to go to a doctor, then your samples are all sent to these centralized pathology labs. Like if you're in a remote area, these samples, they're hiring planes to fly your pee around because they can't do it at the doctor's place because all their things are centralized. So we're currently working with medical professionals and stuff, hopefully in the long run. So once we've got this prototype, we can give it over to them and they can do all of the testing in their laboratories. Biofoundry is going to be a PC1 lab, which means that we can test with non-pathogenic organisms there, things that aren't going to cause disease, but once you want to test with the pathogens, chlamydia and gonorrhea, you'll need a PC2 lab, physical containment 2. So that's when we'll be able to partner with medical and pathology groups, send it off to them, and they will hopefully be able to run the tests for us. And that's also good because they're all accredited to run those tests and certify it properly and legally so that the device is all above board, basically. So that's hopefully we're hoping to get it into GPs for point of care testing. You'll be able to get your results within an hour or two, essentially, so same day results, which is heaps better than waiting 3 days and having to go to the doctor twice. But in the long term, it would be really awesome if we could work with the TGA, the Therapeutic Goods Association, who regulate this kind of thing, and with legal bodies to maybe even find a way that we can get LAMP diagnostic devices into people's houses. And if we've got it user-friendly enough that they can't fuck it up, then they can test themselves for STIs. So at the moment, we've got a lovely circuit board design that Matt has mainly been working on. Tom's been helping him. So that's looking nice. We can now send it off and get printed circuit boards back.
It's in China now.
It's in China now.
Yeah.
Nice.
The first one was all hand soldered, so that took like most of the
30
academic
hours.
weekend. So now we can just get it printed, which is super awesome.
Nice.
We're also working on getting like a case that we can 3D print out and optimizing the LAMP reaction, so getting like the best mix of reagents, optimizing it hopefully to work with the contaminants that you might find typically in urine rather than needing to clean up the DNA. And moving forwards, we're looking at different materials and designs for the microfluidic cartridge that's going to sit on top. That's important because we want it to be able to mix the reaction really well, but we also want to be able to be cheaply produced, because while the device obviously is reusable, the cartridge is going to be changed every time you do a new sample. So hopefully you can get the cost of that down to $1 or less, basically, once you're mass producing it. So like the whole device overall, we're thinking will be like under $300, hopefully, to buy the full device, and then each reaction is less than $1, each cartridge is less than $1. So once you've got the initial investment, it's hopefully going to be pretty cheap then to just keep running tests. I already mentioned that, testing with non-pathogens in our lab, sending it to industry partners to once we want to look actually working with the bacteria, and that's where we're at.
Just one thing, it can be used for any other pathogen with the appropriate primers for it.
Oh, yeah.
So we do want this just to be spread around the world and just used by anyone.
Yeah.
The device, yeah.
Yeah. So our initial target was STIs because we think it's an area that could be improved for STI testing. But essentially the device is it amplifies DNA. So if you want to look at an environmental sample and you've got the primers, you know what you're looking for, you can use it for that. If you want to check if there's salmonella in your food, get the salmonella primers, you can look for that. It's really versatile as well. It's probably really useful for like university labs, like when you're learning and you've got like a 3-hour laboratory, you could run this within the lab. Usually they do PCRs over like 2 or 3 weeks when I was studying, so like you could do this in one tube and it would be good.
Be really useful in the whole outbreak.
Yeah.
Are you able to just publish the original paper, the group, that just on the website, on the meetup site?
Yeah, sure.
So effectively, what is that group doing now that effectively published their open source plans?
Yeah. So
what are they doing with it?
I think it was just someone's PhD project.
Isn't that interesting?
And they were like, here's a really cool thing, I want everyone to use it, it's open source. They published like
the whole specifications. They publish the circuit boards. They publish the costing lists.
Okay, so usually with open source, and there are a lot of commercial open source projects, will you and Biohacker Foundry publish the layout, the PCB, and all the other designs that are involved with this?
It's on GitHub right now.
Yeah.
Already is.
Yeah.
Yeah, so it's up on GitHub, and it's all going to be kept open source. Even like the industry partners that we're talking to, they're really happy to keep it open source.
Absolutely.
So
That's what
it's
it's all about.
yeah, really cool.
Okay, cool.
The tricky part is that because it has to go through a health professional, and NATA have to basically say, yes, you can test on this for that notifiable disease. Unless you get accreditation, it's illegal to use it, right? So even if, say, the industry partner, they did all the accreditation, had a thing, no one could really steal it or do it because they need the accreditation, which no one's going to just walk up and for if someone else has already done it. So
That's,
like there's protection to the industry partner, like
as long as the it's all sort of open
yeah,
to the
but
community.
you wouldn't stop someone with a nice instructable going and building it at home anyway.
Yeah.
What do you guys think about early cancer detection? Because majority of the cancer biomarkers show up really, really early, and you can probably use your system to detect it fairly early as well.
Yeah.
Come help.
Yeah, no, it could definitely be used for that. I think like we're kind of focusing on targeting the STIs at the moment because that's how we can get through all the testing as well with pathology labs that are going to partner with us. But if there are lamp primers, or if someone makes lamp primers for whatever cancer biomarkers you're looking for, it could be used for that as well. Yep.
What's the production cost per unit for this?
So per unit, it varies obviously depending on whether you're making like a single one as a prototype or mass production. So mass production, the per unit would hopefully be $300 or
No, that's $50.
$50. Okay.
Max 100. Max 100.
50, yeah. So $50 to $100 for making heaps of them. Yeah.
Yeah, 2,000 quantities.
Yeah, in quantity of 2,000 or more, would be $50 to $100 per unit.
Yeah. Are you the pioneer in the technology of LAMP, or are you the pioneer in actually creating the machines? Because I think I heard about LAMP Diagnostics before. It's actually a very good technology for detecting diseases like malaria, for example, tuberculosis. So if you actually develop this and then sell it or implement it in developing countries, you can do controlling of the disease. At the same time, you eliminate the disease, which means you save thousands of lives.
Yeah, yeah. People are really excited about using like lamp amplification to detect malaria and stuff. Mainly because it's fast, it's really robust, so it's really good for use in the field. Like if you're in a developing country or a really remote area, and also because it can be done a lot cheaper. You need like smaller sample volumes, which makes the whole reaction cost cheaper even. And in like the device in this paper from Myers et al., they had actually also incorporated like a GPS unit into their thing, so you could also do like geo tagging and stuff of where you're getting results. That wasn't as important for our design because we're going to be putting it hopefully into doctors' offices. But if for other uses like that, you can put GPS units in and stuff as well.
So you are actually the pioneer in this tool, isn't it? So this tool of yours is actually unique compared to the others. There's nothing like this in Australia.
Yeah, not at the moment. I mean, in Australia at the moment, all the pathology labs are still using like old school PCR amplification stuff.
We only just switched to PCR in some tests. Like we're still, a lot of stuff is still culture. Like so we just did a big study in Queensland on the difference between PCR and culture over 6 years for like with the government there, and now they're like, yeah, PCR is better. We're gonna fund that instead. It's like they're really slow to move on all this kind of stuff.
They're like 20 years behind.
Oh yeah.
But it's effectively a snapshot of someone's PhD.
Yeah, I think so.
Yeah. It's just you'd like to mass produce it.
Yeah, basically take it further and actually make it accessible to people.
So who's the PhD student? Are they over in the Netherlands or?
They're in a university in California.
Okay.
They had on their paper in their abstract that one of their goals is for technology like this to proliferate. I tried to email them, didn't get a reply. I looked up their current projects. They're doing really cool stuff.
Okay, good. Thank you.
Awesome.
What's the percentage of diagnostic sensitivity and specificity of this?
So it varies, I guess, depending on what your primers are, what you're looking for, but it's more specific than traditional PCR. So there's been studies that they've detected E. coli down to 10 copies of DNA, which is like nothing. That's crazy. There's also, I think, I always remember the E. coli 10 copies, but yeah, basically really specific. There's actually there's more problems with false positives, if anything, which is something that you've got to work on controlling for. Yeah, but it can detect down to really tiny amounts of DNA.
How do you, like if someone makes one of these, how does the first person calibrate it and say, oh, this one's working or this one's not working?
That's something we're working on now. But we're going to get, I guess, infuse standards and known quantities of DNA. You've also got to calibrate, I mean, like between all the wells, because LEDs can emit light not exactly evenly, so you've got to then figure out how you can calibrate to get each well normalized between the samples, as well as then getting standard curves of the DNA of what you're looking for, so that you can then quantify exactly how much you've got.
The process of developing a control protocol. So every diagnostic machine that's ever gone into a lab to do routine testing has like sets of controls, and they'll have we have fridges full of these controls that we get from regulatory bodies that go, you have to put your machines through these every few days or whatever to make sure everything's working. Theranos just got their ass kicked in the US because their quality control was off and they still rent patient data. So this stuff is really routine, and you just got to develop it for that thing, and industry partners will provide a lot of stuff for us.
That's a couple of questions. What was the original name of the project? And two, how do we help support the cause? And is there a website we can go to fund, or is there something that we could do to volunteer?
Well, we're having meetings about every month, which are advertised through Biohack SID. So if anyone wants to get involved, they can join that. We've also got there's a Slack if people want to join up. I'll put the details on this event if anyone wants to be added to that, so they can help work on it. It's really good actually to have a really diverse group of people, because we've got a whole bunch of different stuff. We need to work on the actual reaction components, what's going into it, but then there's also the circuit board, the software, and the microfluidic cartridge. So expertise from a whole range of areas would be really useful if anyone wants to help out. We don't have an actual website yet, but we should make one.
Someone can help out.
Yeah, I don't know website design, so yeah.
What's the problem? What's the issue with this? Because nobody's producing it. You're the first one, as I.
There are.
Yeah.
There are
people
because it's more accurate, it's cheaper, is smaller, is let's say better in many ways. But people prefer to put.
I guess it's changing from the old system. It's maybe people are worried about having to invest a bunch of money to get a new system when they have an old one, but yeah, we kind of also came across this paper and we were like, why is no one making this yet? This is really what?
Mm-hmm.
Yeah. Yep.
Is there any kind of a competing IP around this from competitors or similar?
Not that we know of. We are still searching, but as far as we know, I think not yet.
Having the whole base project open source helps too.
Yeah. And that was released 3 years ago now.
Yeah. Yep.
When you say no one's making it, are you referring to Australia or the world?
Australian one, yeah. Sort of more, I guess because they're targeting an Australian market to begin with, so we're really focusing on
So does
what's
that mean
available
that there
here.
are people in other countries doing this as well?
Of course there would be.
Not outside of research labs.
I disagree, because if the doctorate is if the publication's out, there are groups in tandem, in parallel, that have exactly the same interests. It's just you may not know.
Yeah, and I mean there are research groups who are writing papers on using
Absolutely.
LAMP for detecting a bunch of stuff, but it's whether they're also deciding to commercialize that.
One way is actually to just go on a forward citation on the paper, and then you can actually just keep track academically.
Yeah.
Yeah.
I can show you a bunch of that already exist, but their target market isn't places like Australia or the US because of regulating bodies like the FDA or NADA. It's near impossible to get.
So that's probably
The reason we have an option here in Australia is because we have companies that are willing to pay through the accreditation process, which they don't have contacts to or can't access. So they're targeting places where they don't have that regulation. And there are a number of different mechanisms. There's one which is attached to a iPhone 5C specifically, and only that model, that is literally a thermocycler that like hugs the phone, and you put your PCR in the top, and it does that. And they've talked about maybe using LAMP as well. But I mean, like, you know, those things are out there, but they're not anywhere near FDA either, or any of these things, because it's just too difficult. It's too you know, as a research device, like it's just not feasible for them at all. But what we want to do is give that kind of access, that kind of tool, which you say, why isn't anyone doing it? We want to give that to a place like Australia or the US where we can take advantage of that, and we can, if you can get it through the accreditation in Australia, you can get it through anywhere. So.
Oh really?
Yeah.
That's interesting.
Well, the people that pioneered a lot of the PCR stuff in labs was in Australia. The guy that helped do it is on our genetics team.
Are you saying that the FDA then will sequentially just immediately follow on from?
Not immediately follow on. They're doing their own thing, but like having any kind of backing anywhere that's like not dodgy. And Australia is classified as not dodgy. So, you know, it helps a lot. Yeah, like, and pathology specifically, we have NADA and the RCPA, it's like the something Pathologist Association. So yeah, and they're a pain in my ass every day. All the stuff they're trying to fix in labs, like it comes down to like how do you take down pencil notes on a fridge temperature gauge, like they're nuts. So if we get through them, FDA's.
Any more questions? Oh, good.
Yeah, so before we all go to the pub, there was one last thing I thought we should do is if anyone's working on a project or has an idea that's related to biohacking or to Ethereum, if you want to come to the front and just give like a quick 1 minute pitch so people know who you are, know what your idea is. Literally 1 minute, like my name, this is my idea, these are people I'm looking for, and then we can kind of like find each other at the pub afterwards. And 'cause I know a lot of people are looking for devs, particularly in the Ethereum group. I'm not sure about the biohacking group, but yeah, anyone who wants to come up? Yep, awesome. Line up.
All right. Hi, my name's Tim. We set up a company called Veridictum, and we are creating a smart ownership and distribution platform for video content producers to protect them from theft and piracy, and to create a new range of distribution models. We're about to go into Blue Chili, and we've got a seed round coming through, and we're talking to film industry people for the next rounds of funding. Interested in devs and anybody that's got any form of interesting technology. Really interested in having a chat.
Hi, my name's Hugo. I'm from BitTrade. We're looking for software engineers. We're doing some cool stuff in Ethereum at the moment. Some greenfield projects. I think it's really exciting. If you're interested, just come and see me after and I'll talk to you about it.
Hey guys, I am Manuel. I work in a startup here in Surrey Hills. So basically we're doing an IoT integration platform. We want to use Ethereum for all the authentication. So if any one of you is interested, come and see me. We're going to be deploying a LoRa, long range network. So it's a kind of like a Wi-Fi for Internet of Things. In Surrey Hills, it's now covering all the city to North Sydney. So it will be pretty cool if you want to come and talk. Nice.
All right.